Hi everyone, we are continuing on with our fourth video in topic 2, the slippery slope of life. So we've already covered what is life, how are prokaryotic and eukaryotic cells different, and how did eukaryotes develop. So now we're going to look inside our eukaryotic cells and describe all the components of the cells. So our learning objectives, by the end of this video, you should be able to describe the structure of eukaryotic plant and animal cells. So we're not really necessarily going to cover fungi or protists in detail. So it's basically, there's similar overlapping components, but we'll focus mainly on plants and animals. And then we'll summarize the functions of the major cell organelles. So before we get started, let's just review the differences between prokaryotes and eukaryotes. So if we recall from a couple videos ago, or a couple sections ago in our textbook, prokaryotes, they are a lot smaller than eukaryotic cells. So prokaryotes, they're about 1 to 5 micrometers in length, whereas eukaryotic cells, they can range from 10 to 100 micrometers, even larger than that. Our prokaryotic cells, they tend to be very simple in structure. There's not much going on inside of them. They don't have any organelles. They don't even have a nucleus, so their DNA is just found in the nucleoid region. Whereas the eukaryotic cells, they are very complex. So they do have a nucleus. They do have different organelles inside, and those organelles all have different functions. And then as far as the types of our different organisms that have prokaryotic versus eukaryotic cells, we have our domains of life. So bacteria and archaea are two domains of life that are fit under the prokaryotic cell category. The third domain of life is eukarya. So they're made of, eukaryotic cells, makes sense. And the 4 kingdoms, or protists is not really kingdom classification anymore, but the 4 classifications found in domain eukarya are plants, animals, fungi, and protists. So again, we're going to focus mainly on plants and animals when we talk about the eukaryotic cell. So let's take a look at these two pictures here. So first off, which one is the animal cell? Which one is the plant cell? So right off the bat, go by color. Okay, so plants, they are green because they have chlorophyll, which is found in their chloroplasts. Okay, so plants have the chloroplast. So this over here is our plant cell, and this over here is our animal cell. So what I want you to do is think about which structures does the plant cell have that the animal cell does not. So look around, see what the animal cell has and what the plant cell has. So one thing I can see right away is the chloroplast. Okay, so animals, we don't photosynthesize, right? So we have no need to have chloroplasts. Okay, so plants, they have a chloroplast, we do not. Another thing that we don't have is a central vacuole. We might have little vesicles and maybe even a vacuole, but we don't have a central vacuole. Okay. We don't have a cell wall. So our cells, the boundary is just this, this plasma membrane, which plant cells also have, but outside of their plasma membrane or cell membrane, they also have a cell wall. And then because they have a cell wall, they also have something called plasmodesmata, which are little channels that connect two plant cells to each other. Okay, so we can see there are, plants have the chloroplasts, they have central vacuoles, they have cell walls, and they have plasmodesmata, which animal cells do not. So let's flip it around. What structures does an animal cell have that a plant cell does not. Okay, so we have cytoskeletons like they do, plasma membrane like they do. Do they have lysosomes? They do not. Okay, so they have plastids which store pigment. We don't really have that. Lysosomes, we have lysosomes that contain little digestive enzymes, so they don't have those. So that's one characteristic that we have that they do not. Anything else? That looks like about it. Yeah. They don't really, other than that, there are a couple others that are not mentioned here, but so we don't really need to know it for this purposes of this class. So the only one that looks like it's different is the lysosome. Oh, there it is. I was wondering what it was. We have something called centrioles, which they have something else. They have they actually have centrosomes, but it's not listed here. Okay, so we're actually going to go into more detail of some things that plant cells have that animal cells do not, and vice versa. So let's get into the different classifications or different types of components of the cell. So we're going to start out with the plasma membrane. So remember, the plasma membrane, all cells have this. It doesn't matter if we're talking about a prokaryote or a eukaryote. So all cells have the plasma membrane. So when we take a look at the plasma membrane, just take a look at it. It's actually pretty complex if you look at it. So our plasma membrane is composed of a phospholipid bilayer. So phospholipids, so basically it's phosphate and lipid. So it's we have a phospholipid bilayer, so there's 2 layers of phospholipids. And we'll go into more detail of what that means later on in the term. So it actually is going to create this specialized semi-permeable membrane because the phospho part is going to be something called hydrophilic. So hydrophilic, philic means liking, hydro means water. So it's actually going to be, okay with water. Hydrophobic would be the lipid part. So think of, lipids, fats, they don't like, they don't mix with water. So they're hydrophobic. Phobic means fearing. So the lipid part, the little tails, they're facing inward to each other and they don't like water. So we have part of the phospholipid bilayer is water loving and then the middle layer here is water fearing. So that's kind of interesting how it's set up, and we'll talk about more why it's like that in a later unit. And then we also have other things. We have these little proteins embedded within. So we have like little protein channels. We also have proteins on the outside of the membrane. Okay. We have like cholesterol that's embedded within the membrane. We have little glycoproteins. So glyco means like a carbohydrate, so that's attached to it. So, it looks like a very complex membrane here. And so what's the purpose of it? Well, it separates the inside of the cell from the outside of the cell. Okay, so the inside of the cell is kept separate from the outside of the cell. So the internal components of the cell is separated from the external environment. And like I said before, it's very complex, and the reason why it's complex is because it's semi-permeable. So certain things can easily move into and out of the cell through either active or passively, move through, but other substances can't. So, certain things cannot go through. It will not pass through easily. Okay, so certain things can pass through, maybe through a protein channel, or maybe even squeeze through the phospholipid bilayer. but other substances can't, and so it's semi-permeable. So that is our plasma membrane. And also you'll find it called the cell membrane, same thing. Next up, we have the cytoplasm. And remember, the cytoplasm is also a basic unit of the cell. So all cells have this. Now, your textbook doesn't really describe the cytoplasm, so other than it being just this jelly-like substance, so I'm not going to describe it either. I'm just going to go with what the textbook describes. So suspended within the cytoplasm is the cytoskeleton. So think of cytocell, skeleton as, well, what does your skeleton do? It kind of provides structure to the cell. So our cytoskeletons, or the cell cytoskeletons, we have three components to it. We have microfilaments, intermediate filaments, and microtubules. And these are ranked from smallest to biggest. So microfilaments are the thinnest, intermediate filaments are kind of intermediate in size, and then the microtubules are the thickest. So the microfilaments, they help move cellular components. So for example, when we're talking about like cellular division, they kind of, make up the mitotic spindle and stuff like that. The intermediate filaments, they provide structural functions. So, that's basically all you need to know about them. Microtubules are the thickest, so hence the strongest, and they guide organelle movements. So think of if we're transporting a vesicle from one side of the cell to another, they'll hitch a ride along one of the microtubules. So the cytoskeleton is, basically the structural support within the cell. The skeleton of the cell. So within our cell, we have something called the endomembrane system. So think of endo as within. So within the membrane system. So the endomembrane system is a group of membranes and organelles that kind of work together, and their purpose is to modify, package, and transport lipids and proteins. So typically they are transporting those lipids and proteins for export out of the cell. Okay, so especially like, for example, in our bodies, cells secrete things. So think of in the pancreas, for example, we have our pancreas secretes a hormone called insulin. And so the endomembrane system within our pancreatic cells is going to be producing the hormone insulin within the cells to be secreted into our bloodstream to help regulate blood sugar levels. And so, that's something that's going to be happening, which involves a number of different organelles. So we have the nuclear envelope that's involved. So that's basically surrounds the nucleus. The lysosomes, we'll talk about those. The vesicles, the endoplasmic reticulum, and then the Golgi apparatus. So we'll talk about each one of these that's involved with the endomembrane system. And that endomembrane system modifies, packages, and transports lipids and proteins. So let's start out with the nucleus. Now I like to think of the nucleus as the boss of the cell, okay, or the CEO of the cell, or whatever you want to think of it, because it houses the DNA. So this is going to be where our genetic information is. And it directs the ribosome, it basically directs the cell to make proteins, okay? It directs ribosomes and protein synthesis. Now, when we look inside of our nucleus, we can see the DNA. Now, DNA, it's not just DNA. It's wound into chromatin. So chromatin is DNA plus protein, okay? It's just because the DNA is really, really long. And so what happens is the DNA is wrapped around these types of protein called histones. And so, it's kind of like a spool, like if you're going to have thread, you're going to have a spool of thread instead of just it loose, because if it's loose, it's going to be bigger than if you put it on a spool. So that's why it's kind of spooled up into this little, around these proteins, and to form chromatin. And then the chromatin is kind of wound up into a chromosome. So a chromosome is just like a linear piece of DNA made of chromatin. So for us humans, we have 20 or 46 of these chromosomes for us humans, and it varies from species to species. So not all organisms have 46. Some organisms have more, some have less. And it doesn't have anything to do with complexity. So for example, some plants have like 5,000 chromosomes, and that doesn't mean that they're smarter than us. All right. If you can take a look at the center of our nucleus, you see this dark purple splotch, and that is called the nucleolus. Okay, so the nucleolus is basically, it contains the ribosomal subunits. Okay, so remember, ribosomes are little protein factories. So this is the area that has the components to make them. Okay, so it contains something called rRNA. So R stands for ribosomal RNA. So ribosomes are made of ribosomal RNA, basically. Okay, so it contains little subunits. So this is where that is made from. Okay, so if we look inside the nucleus, we have our chromatin made, that's housed on our chromosomes, and then we have our nucleolus that has the rRNA, okay, that makes the ribosomes. And then we have our nuclear envelope that surrounds it. So our nuclear envelope is a double membrane. And if you look at our double membrane, you see there's little gaps. So those are the nuclear pores. And that allows for things to move through. Okay, so for example, when we are sending messages from the nucleus out into the cytoplasm, it can pass through these nuclear pores. Okay, so that is the nucleus, the boss of the cell. Next, we have the endoplasmic reticulum, otherwise known as the ER. So the ER is a series of interconnected membranous structures within the eukaryotic cells that collectively modify proteins and synthesize lipids. So you can see where the nucleus is relative to the endoplasmic reticulum. So it's basically connected. So the nuclear envelope basically the endoplasmic reticulum forms off of that nuclear envelope. And we have two types of endoplasmic reticulum. We have the rough ER and we have the smooth ER. Now the difference between the two is pretty obvious. Okay, you have these little ribosomes attached to the rough ER, and the smooth ER does not have them. Okay. Now remember what the ribosomes do. They're the little protein factories. So you can think of the rough ER as they're the ones that have the protein factories on the surfaces of the membranes. So they're engaging with protein modification as well as phospholipid synthesis. So they are basically making and modifying proteins and phospholipids. The smooth ER, notice there's hardly any, if any, ribosomes. Okay, so they're not making any proteins, right? So instead, they are synthesizing carbohydrates. They're making the lipids. So whatever lipids are made here can go through the rough ER to be modified into phospholipids and things like that. They can make hormones. So steroids are a type of lipid. So steroid hormones. They can also serve other purposes besides making things. So they can detoxify certain chemicals. So they can also metabolize alcohol, and they can store calcium ions. Okay, so there's a number of different functions of our endoplasmic reticulum. So they can manufacture a number of different molecules, as well as the smooth ER can help remove toxins and store calcium. Okay. And that's part of our endomembrane system. So our endomembrane system starts with the nuclear envelope, and then here we have the rough ER and the smooth ER. Next, we have the Golgi apparatus. So the Golgi apparatus is detached from the nucleus. So that's an easy way to tell the difference between the smooth ER and the Golgi apparatus, because they kind of look similar. They just look like flattened membranes. But the smooth ER will be attached to the membrane that's attached to the nucleus. The Golgi apparatus will be detached from that. And so the Golgi apparatus, I think of it as like the shipping center. Okay, so it basically it will receive the whatever is manufactured. I think of this as like the factory floor. Whatever is manufactured by the rough and smooth ER will get transported within some sort of transport vesicle into the Golgi apparatus. And once it's transported here, that transport vesicle will fuse with the membrane of the Golgi apparatus. And here it kind of gets sorted and tagged and modified and packaged so that it can then be travel again through a different transport vesicle where it can then get released from the cell. Okay. So think of the Golgi apparatus as the shipping center of the cell, okay? So we have the boss of the cell up here, the nucleus, and then we have the factory floor making the products, and then we have the shipping center that is, putting everything in boxes and putting the shipping labels on them, and then the transport vesicles are shipping them out of the warehouse, wherever they're going, shipping them out of the cell. All right. And you can see that the Golgi apparatus, it actually has two sides to it. So it has the cis face and the trans face. So the cis face is the side that's facing the rough ER. So it's the receiving end. And then the trans face, it's going to be the end that is transporting the final product out of the cell for distribution. Okay, so that's our Golgi apparatus. All right, and then we have some other parts that are involved, such as our lysosome. So our lysosome is a special type of organelle that is kind of like the garbage disposal of our cell. And if you recall from a few slides ago, it's only found in animal cells. And it contains these digestive enzymes. So it helps break down things that shouldn't be in the cell. So it breaks down like proteins or polysaccharides, which are little carbohydrates, any lipids, nucleic acids, or even things that are kind of done. So like if there's some mitochondrium that's just worn out, it can engulf it and just break it down and get rid of it for us. So it's kind of like the garbage man or janitor, or even the security guard. It'll get rid of like bacteria that has somehow entered the cell. So lysosomes, they're garbage disposal part of the cell. And they do that with their enzymes. So they have specialized hydrolytic enzymes, which are at a lower pH. So since they have a lower pH, it allows them to kind of break down substances. So you can see how they could be kind of a security guard. So they're going around, and if there's a bacterium, they can kind of engulf it and then break it down. Okay, so here's an example. This is a macrophage. It's part of your immune system. So it kind of engulfs it and then breaks it down. All right. A couple other types of components that are found in cells are vesicles and vacuoles. So the only difference between a vesicle and a vacuole is the size. So vesicles are small, vacuoles are large. Okay, so the vesicle is a small membrane-bound sac that functions in cellular storage and transport, whereas the vacuole is a larger membrane-bound sac that functions in cellular storage and transport. Okay, so the other difference besides size is that vesicles, they can fuse with the plasma membrane and the endoplasmic and reticulum and the Golgi apparatus, whereas the vacuoles don't. Okay, they don't fuse with other cellular components. So in the previous slide, let me go back. We can see like this transport vesicle came off of the Golgi apparatus, or excuse me, the Ruffyart, and is fusing with the Golgi apparatus. Here's another transport vesicle that comes off of the Golgi apparatus and fuses with the plasma membrane. So these are vesicles. These are not vacuoles. Vacuoles do not fuse with other organelles or plasma membranes. Okay. So that's the difference between the vesicles and the vacuoles, as well as their size. But they are involved with transporting things as well as cellular storage. Okay. And then our ribosomes. So we're already familiar with ribosomes because all cells have ribosomes. Those are our little protein factories. So they carry out protein synthesis. Now, you don't really need to know anything on that slide, especially since it looks like very complicated and even in, not really readable. You can't even read it anyway. But basically, our ribosome is involved with protein synthesis, and it's going to be making proteins. So here's an example of an amino acid sequence that is going to be eventually become a protein. So it's going to be using our blueprint. This is a messenger RNA strand, and here is our ribosome made of our RNA, ribosomal RNA, to do that, to make our little amino acid sequence. Now we have two types of ribosomes. We have ribosomes that are just floating freely throughout our cytoplasm of our cell. So those ribosomes, they make proteins to be used within our cell, okay, because our cells, they need proteins to build things within the cell. And then we have the ribosomes that are attached to the rough ER. So those are what we call the bound ribosomes, okay? So they are our protein factories. And then moving on to other components of the cell, we're already familiar with mitochondria and chloroplasts from our previous video. So learning more about them, mitochondria, so those we call them the powerhouse of the cell, because they can convert energy from an unusable form to a usable form. So they take cellular energy from our nutrients from our food and convert it, chemical energy from the nutrients from our food and convert it to the energy that cells use, cellular energy, which is ATP. So they do this through a process known as cellular respiration. So we'll learn about cellular respiration in another unit. But this is basically where it occurs in our cells, in the mitochondrium. And we've already talked about our mitochondrium has its own DNA and its own ribosomes. Okay, so we'll discuss cellular respiration later on, but here is that organelle inside the cell. So that's the site of cellular respiration, where we produce cellular energy in the form of ATP. So that's our powerhouse of the cell. So this is providing energy for the cell. And then we have some components that are only found in plants and some components that are only found in animals. So one of the components that's only found in plants would be the cell wall. So the cell wall, of course, we don't have that, and that's a good thing because we're very flexible, mobile creatures. So we don't want that rigid structure that surrounds our plasma membrane. But plants, they like it because it helps protect them. So cell wall is a rigid covering. So for plants, that rigid covering is made of cellulose. For fungi, it's a different type of component called chitin. So we'll talk more about cellulose in the next topic, topic 3. But cellulose is a type of carbohydrate, and you might be familiar with it because it is dietary fiber. Okay, so think of dietary fiber. So plant cell walls is basically dietary fiber. And what that does, that cell wall, the reason why plant cells have it, so here's its cell membrane or plasma membrane. So it's outside of the cell and it protects the cell. Okay, so it's a form of protection. It also provides structural support. Okay, so it allows the plant to, be able to be upright. Okay, so it defies the laws of gravity because it can grow towards the sun. And it also gives shape to the cell. Okay, so it provides, structure, shape, and protection for the cell. Also, plant cells have chloroplasts. So of course, we don't have chloroplasts because we don't photosynthesize. So plants, they can't just, eat like we do. They don't have mouths, they don't have digestive tracts. So they have to bring in their own source of energy. So they have to do that through their chloroplasts. So chloroplasts, they are the site of photosynthesis. So inside the chloroplasts, they have these little things called thylakoids. Thylakoids, inside that, they have a pigment called chlorophyll. So that chlorophyll is a green pigment. And that pigment is able to absorb light energy from the sun. And that energy source allows the plant to convert carbon dioxide and water into sugars, chemical energy. And then it also produces oxygen from that process. Okay. Now plants, they do have mitochondria as well. So that way the plant can just take the sugars that it produced and transport it over to the mitochondria through a little transport vesicle, and then it can produce cellular energy from that. Okay, so plants, they don't need us, we need them. Okay, so they are self-contained. And then just like we talked about earlier in the last video, chloroplasts have their own DNA and ribosomes, just like the mitochondria. All right, and then another thing that our plants have that we don't have a central vacuole. So inside the plant cell, there's usually present a large vacuole. So that organelle is a storage compartment. So it basically provides a place to store things for the plant. So it could store pigments. So think about in the flower, when the flower is in bloom, it can have a color pigment in there, so to produce a pretty color, or a toxin perhaps, or it could store water if it's like in a leaf, because we don't know next time, if it's a house plant, we don't know the next time you're going to water, so it needs to store water for a while. And so it could provide for water storage or, storage for food. It could be the site of macromolecule degradation, so it could, store starch in there for later use. So it could be, basically it's a storage place for other things. Because plants, they cannot just get up and walk around and go find a resource. So they have to rely on the environment, and so they have to store things away for a rainy day, I guess I can say, until the next rainy day. So that's why they have the central vacuole. All right, so we talked about the things that plants have that we do not. So the cell wall, the chloroplast, and the central vacuole. So now we're going to talk about something that we have that they don't have. And since we don't have a cell wall, that gives us room for something called the extracellular matrix. So this is present in plant cells, but not, animal cells, but not plant cells. So outside of our animal cells, we have this extracellular matrix. So here's the inside of the cell down here. This is just outside the plasma membrane. So we have this extra matrix, cellular matrix stuff. So this is outside of the cell. It's secreted from the cell, and it's mainly, made of like collagen fibers, glycoproteins, and these other things called proteoglycan complexes, which have carbohydrates, proteins, and polysaccharides involved. So what this does is it can hold cells together as tissue. Okay, so remember our organization of life. We have the the cells, multiple cells together can form tissues. Tissues can form organs, organs can form organ systems, and so forth. So the cells held together to form tissues, that holding together is from this extracellular matrix. So the clinging together is due to this extracellular matrix. It also allows our cells to communicate with each other. Okay, so it allows the cells to be able to communicate. Give each cell what they need. Transport things back and forth, stuff like that. It also provides mechanical protection and anchoring for cells to in the tissue. So we can anchor the cells to each other and provides that mechanical protection. So it kind of zips them together. So that's that extracellular matrix. All right, so here is our review question. So which of the following is found both in eukaryotic and prokaryotic cells? Is it the nucleus, the ribosome, the mitochondrium, or the vacuole? If you chose the ribosome, I would agree with you. So that's one of our 4 basic components of a cell. The nucleus is found in eukaryotic cells. Mitochondrium found in eukaryotic cells. Vacuole is found in eukaryotic cells. Central vacuole, plant cells, which is a eukaryotic cell. All right. And then, by the way, if you get a chance, go check out your Adigia textbook. At the very end of this section, there is a great chart that has everything there. Okay, so it has the components of prokaryotic and eukaryotic cells and their function. It gives the cell component, the function, and then if it's present in prokaryotes, if it's present in animal cells, and if it's present in plant cells. So this is a great resource to use to help you study. Okay, so that's it for our 4th video on topic 2. So by now you should be able to describe the structure of our eukaryotic plant and animal cells, be able to tell us what animal cells have that plants don't, and what plant cells have that animals don't. You should also be able to summarize the functions of the major cell organelles, such as the nucleus, the endoplasmic reticulum, the Golgi apparatus, and so forth. So our next video, we're going to get into what are viruses, and are they considered cells? And if not, why not? So I'll see you there.